Hey everybody, welcome to the Lab Insider. This is a new series, which is going to be a MedLab lecture series. And essentially what we are going to do is we are going to look at different aspects in medical science and we're going to go through it as a lecture. I'm going to be doing tutorial videos. This is a side segment from my discussion segment of the Lab Insider. Today's topic is going to be hematology cell types for differentials. So essentially we're going to be talking about diffs and we're going to be talking about cellular morphology and basically what that entails and what you need to know about cellular morphology, how you can identify various types of blood cells, red cells and white cells and platelets alike. So everybody, the normal erythrocyte, what it should look like, is very similar to this image here. It's essentially a disc, which is about 7 microns in length. The erythrocyte has a central pallor, which can be seen in the center of it, which is a bioconcave part of the disc. And now the central pallor should take up about 1/3 of the whole surface area of the erythrocyte. So you can see the central pallor kind of dips in. It's where the nucleus of the cell has been actually pitted out through maturation processes. Okay, and that's really what you can see is that kind of concave part. So that's why the erythrocyte is considered to be a bioconcave disc. So this is essentially what a normal red blood cell is supposed to look like. The central pallor, obviously, in a peripheral blood smear appears more whitish, as the erythrocyte appears more pinkish, depending on the stain. Most of the times it's an eosin stain. So essentially, that is an image of a normal red blood cell in terms of size and morphology. So this is essentially a normal peripheral blood smear, and normal erythrocytes, and how they're spread out amongst the whole slide, how they're supposed to look, what we're supposed to see in a normal smear. If we take, for example, the cell in the center, this cell has a 1/3 central pallor, which is exactly what we're looking for. It doesn't take up the whole surface area, and it doesn't take up very little. It's essentially there. You can see the slight discoloration. The cell is actually not um, darker stained. It's not very deeply stained, which is what we want to see. Essentially, what we call this is normocytic, normochromic, which means essentially normal cell size and morphology and normal coloration, normal stain. Talk about cell size and coloration, we essentially talk about certain values that we can calculate. For example, the MCV value, the mean corpuscular volume. The hemoglobin plays a very big role, as well as the hematocrit as well. I won't be going into MCHC per se. The MCV is in femtoliters. It's the mean volume of the cell. So a normal sized cell like we saw in the peripheral smear is approximately 80 to 100 femtoliters. That is what the MCV value should be. The hemoglobin value for any normal adult, this taken for an adult male, is 135 to about 170 grams per liter, whereas the hematocrit is 45 to 52%, also in an adult male. It's slightly less in females, whereas total blood volume is about 5 liters. So that's essentially very important values when we're looking at a complete blood cell count. We want to know really if the erythrocytes morphology and hemoglobin concentrations are essentially normal. So these would be normal values. Now, when we talk about microcytic anemias or microcytic conditions, we're essentially talking about smaller sized erythrocytes and oftentimes hypochromic erythrocytes with less coloration than the normal erythrocyte. Essentially, what you see is that the central pallor is much bigger. It's not 1/3, it's approximately now 2/3 of the cell's whole volume. And the hemoglobin concentration is essentially a lot less. The cell is also smaller in size and sometimes takes on a variety of interesting morphologies, which we will be explaining after this segment. So if we look at this smear here, this peripheral blood smear, you can see at the bottom corner there's a lymphocyte. And now you know that a lymphocyte is essentially slightly bigger than a normal erythrocyte. It's essentially just a tiny bit bigger than a normal erythrocyte. If we look around the erythrocytes, around that lymphocyte in particular, what we begin to notice is that those erythrocytes are considerably smaller than that lymphocyte. They are very, very tiny. You can also see the platelets around in little purple colors. Those platelets are quite big compared to the erythrocytes themselves. So these erythrocytes are essentially microcytic. They are also hypochromic, so they are predominantly white. They are dominated by a big central pallor in the center. Um, there's just a little tiny ring of pink around, and they are very small if we look at them in size comparison to the lymphocyte at the bottom right corner. When we look at our values, again, these same values being the MCV, being the hemoglobin and the hematocrit, you can see differences in a microcytic anemia. Essentially, what makes it microcytic is the fact that the MCV is less than the normal lower range of 80 femtoliters. There is usually anemia, which is an associated condition with this type of cellular morphology. So the hemoglobin is usually less than 135 grams per liter, and there's usually always anemia involved. And the hematocrit is variable because packed cell volume usually depends on the quantity of red blood cells as well as other factors. So the hematocrit can actually vary. It's usually lower, though, on the lower end of the spectrum. We're going to talk about macrocytosis. Most common causes of macrocytosis are usually pernicious anemia and folate deficiencies. Essentially, what we see here is the cell maturation stage of the erythrocyte has kind of stopped or has been brought to a halt due to DNA replication issues. And essentially, what we have is we have red blood cells without nuclei, but that are considerably larger. You have considerably larger erythrocytes. Now, you can see where the three arrows are. These point to macrocytes, which are large erythrocytes. And if you look at the bottom left corner, you'll see with the nucleus, it's stained in dark purple, that you have that lymphocyte. And remember how I said that normal red blood cells should be smaller than the lymphocyte itself. Well, in this case, these macrocytes, where the arrows are way, way larger than that lymphocyte on the side. So that's just to give you an idea of how macrocytosis looks on a peripheral smear. You see some cells appear to be normal sized, but there are those macrocytes around there. And essentially, that's what you want to look at when you're thinking perhaps this patient has pernicious anemia. I mean, there's other sorts of pathologies. There's other sorts of morphologies that you'll see as well that are more related to white cells and platelets. But because this video focuses on erythrocytes only, when we look at macrocytic conditions, you have to see large cells. And notice how these large cells have very small central pallor, typically smaller than that 1/3 central pallor of the normal cell. And that's basically because these cells could be hyperchromic. They could have a lot of hemoglobin inside. They have a lot of color. They retain a lot of color. And because of that, they tend to not have as large central pallors. So that is essentially a microcyte. So essentially, when we look at MCV and hemoglobin and hematocrit values, the MCV in a macrocytic condition with macrocytic cells is always going to be greater than 100 femtoliters, so the mean corpuscular volume is actually larger than that of a normal erythrocytes. Hemoglobin and hematocrit, I won't really go into the details about it. Hemoglobin could be low depending on whether anemia is present or not. It really depends on the patient's condition. Hematocrit, like before, could be relatively unchanged, could be lower, maybe higher, depending on the cell count, the red cell counts. So we won't really look at those two values, but the MCV is definitely an important value, and you'll see that it's definitely greater than 100 femtoliters without a doubt. So now we're going to look at poikilocytosis, so poikilocytes and their morphologies. Essentially what poikilocytes are is they are abnormally shaped red blood cells. You'll also hear the word anesiocytes. And basically, we're going to take a look at some of the poikilocytes or abnormally shaped erythrocytes that you can find with certain conditions, and to look out for them when you're actually doing a cell differential. First poikilocyte we're going to look at is the target cell. Why is it called a target cell, might you ask? Well, it's called a target cell because it essentially looks like a target, like a bullseye. And why does it look like a target? Well, if you look at it on a 3D axis, it's essentially bell-shaped. Usually target cells are come out with their poikilocytes that are present with anemias as a main condition. And essentially, because of that lack of hemoglobin, structurally these cells are a little more fragile, and essentially what happens is they kind of cave in and they flop over into each other, so they assume this sort of bell shape, which is why you can tell that the central pallor in the middle is more like a ring of white than really an actual central pallor like a normal erythrocyte. And they're usually microcytic, sometimes not, but essentially the main take-home message is that you're going to see a cell that looks like a target. Think of the target logo. That's essentially what a caudocyte looks like. Um, Basically, remember to always correlate your findings. So if you do see target cells or caudocytes in a peripheral smear while you're doing a differential, correlate that with the actual MCV value, correlate that with the hemoglobin value, because a lot of the times there's going to be signs of anemia, or there is going to be anemia, microcytosis, hypochromia. These cells are usually associated with hypochromia, okay, with less concentrations of hemoglobin in the cell. So always correlate those two findings. Never assume that just because you've seen target cells or caudocytes that there is no anemia or that there is no condition. There should always be some sort of underlying condition, and they are present with a wide variety of anemias: iron deficiency anemia, genetic anemias like beta thalassemias, alpha thalassemias, sickle cell anemias. Whether it's the trait version, so whether you're a carrier, or whether it's the disease, these cells are present just at various different quantities, and various different shapes and styles, but essentially the main take-home message is that it is a target. So whether you say caudocyte or target cell, remember when you see this that that is exactly what it is. Next poikilocyte we're going to be looking at is the elliptocyte. As the name suggests, an elliptocyte is an ellipse or an elliptical type cell. It is oval, it's kind of stretched out, sort of like an oval. In this case, they assume a cigar shape. They're very common in hereditary elliptocytosis, in which the peripheral blood, in the peripheral blood, 40% of the cells, of the red blood cells, are elliptical. They are elliptocytes. That's the shape. It's not a very, how should I say, it's not a very dangerous condition. It is a genetic condition. It is still, however, a type of anemia, or may actually present with symptoms of anemia. But essentially, that is what elliptocytes are. They're cigar-shaped red blood cells that have been elongated. This is normally thought to be due to membrane, the cellular membrane's integrity, and it sort of lacks that kind of integrity, and the cell ellipses in various different ways. So when you see that, essentially try to correlate the results and think to yourself, well, these are elliptocytes. They look like pencils or cigars. And that's essentially what they are. Now, very similar to elliptocytes, we have spherocytes. Instead of being ellipses, they're, as the name suggests, spheres. They are associated with microcytic anemias. They are always microcytic cells, so they are very small and spherical shaped. They are, however, hyperchromic, which means they are sort of very concentrated with hemoglobin due to their small size. They don't tend to have central pallors. They are not bioconcave discs. In 3 dimensions, they are actually spheres. That's why we see them kind of darker stained and without a central pallor throughout any of its surface area. Spherocytes are present in a variety of conditions, a variety of anemias. If you're missing a spleen, spherocytes may be present. Um, that's as far as it goes. When you see very small cells that are spherical, they're typically indicative of being spherocytes. One thing I would also like to mention about spherocytes is because they're related to a membrane defect, their osmotic fragility It's also less, so there's a whole thing called an osmotic fragility test, which I could talk about in another video if you'd want me to. But that in itself is more diagnostic, so we won't be talking about that right now. Now, very quickly, for purposes of speeding up the video, we have dacryocytes, which are teardrop cells. You can see where the arrows are. They're shaped like teardrops, also present in a variety of anemias like sickle cell anemia. I won't get into the details. When you think about that, when you want to think about the name, think dacryocytes. So cry, teardrop, dacryocytes. Now, Acanthus sites have a sort of spindle-like pseudopod projections coming out of them, kind of sticking out all over the place. They could also be called burr cells because, I don't know, the scientists just thought it was cool. Burr. Anyway, echinocytes are very similar. Sometimes they are artifactual. In many cases, acanthocytes are not, and they are associated with certain forms of anemia. Uh, so yes, these are acanthocytes. Lastly, we have sickle cells, also known as drepanocytes. Essentially, sickle cells are part of sickle cell anemia. What is sickle cell anemia? Well, the cell's hemoglobin S structure causes them to sickle under lowered oxygen conditions. So essentially, a sickle cell is an elongated, tapered cell that's kind of spear-like, and it takes the form of a sickle. Think hammer and sickle, Soviet Russia, whatever you want to think, whatever helps you remember that. These are actually very simple to identify. Remember to always correlate your findings again with the presence of hemoglobin S, and also other forms of anemias. There are many types of poikilocytes in sickle cell disease and trait. However, the sickle cell or the drepanocyte is the most common. So that's it for this episode of the Lab Insider, guys. This was part one of our lecture series. This one was on cell differentials pertaining to red blood cell morphologies. The next video that I will be putting up is going to be on white blood cell morphologies. That's where it gets a little more complicated, but nonetheless, it should be super, super interesting. So, if you have any questions, message me. To all students who are watching this lecture series. This lecture series is made not only for text, but also for students who are learning. Feel free to ask me questions. Feel free to request videos, lecture series on anything you might be having trouble with as you're studying the wonderful field that is medical laboratory science. Thanks again. Like always, hit the like button, subscribe. See you guys.